Thank you, Anita, for an excellent introduction. First, I would like to thank the organizers for inviting me here to give this presentation. This is my first visit to Serbia, Belgrade, and I'm quite impressed. Thanks again. Hopefully, I'll come back sometime in future. In the morning, we heard about how to carry out the bioequivalence studies. What are the rules, regulations, and how that needs to be performed. But all that was related to the drug products which are normally absorbed from the body. When you administer it, it goes into dissolution, drug absorption, and you do the pharmacokinetic studies. Well, I'm going to be talking about slightly a different topic, which is the drugs which are locally acting generic drug products. What kind of requirements are there today for the bioequivalency or for a generic product to come for these types of the products? So, no matter who is doing it, what he or they are doing it, the main purpose of the regulators is to make sure that the product is always safe and effective when they pass the regulation, and it is also available to the people. When I use the word available to the people, that means also to the common public, and that is looking into the generic ways, how the generic product could be very easily made available to the public. Before a product is being approved and done and everything, the most important aspect is to remember and have a good manufacturing practice, GMP. In the morning we heard about the GLP and the GCP. They're all good laboratory practice, good clinical practice. But once your product gets approved, again, what's most important? The most important is the product is manufactured under ideal GMP conditions. If you don't have a good GMP conditions, then forget about everything. Your products, whether you do it good or bad or however you do it, is no good. You need to maintain the GMP as far as the manufacturing of the product is concerned. And then again, these are the other different parameters that you try to investigate and study: the dissolution, bioequivalency, the blood level measurement, safety, toxicity studies, and all that. And that's what you heard in the morning from Professor Hinkal, indicating that, when you do the bioavailability studies, you look into the safety and efficacy as an important factor. And when you look into the bioequivalency aspects, you don't go beyond that, but you just look primarily into the bioequivalency of the drug products. Now, coming back to my topic of therapeutic equivalence. What is a therapeutic equivalence, and why do we do that? I know some of the definitions might be the same that you have heard in the morning, but I will try to not read the things but explain to you a little bit. Normally, you have a generic product drug approval. And why do you do that? Because once your product is approved, your goal is to make sure that it is available and it is interchangeable. It is exchangeable with the brand name product, because the generic products are generally cheaper. So that's your goal. And that's why you need what is known as the therapeutic equivalence. But therapeutic equivalence is a combination of two things. One, your product has to be pharmaceutically equivalent, and the second one, your product has to be bioequivalent. So what is a pharmaceutical equivalent product? This is very easy to do because everybody can follow this. It has to be the same active ingredient, the same type of the dosage form. For example, the creams and ointments, they are both different, so it's not the same type of the dosage form. They are different, and when you are comparing it, you have to compare cream versus cream, ointment versus ointment. There is one difference here that's followed in United States and elsewhere, and that is the capsules and the tablets. In United States, the capsules and the tablets are not interchangeable, whereas everywhere else they are interchangeable. Well, there is some reasons behind that, and I have seen some data myself in the laboratories where the in vitro aspects are completely different between a tablet and a capsule. of the same active ingredient. But that's a special issue, and apart from that, it is very difficult very often to justify scientifically as to why the tablets and the capsules cannot be interchanged. Then the same route of administration, that's easy to do. Identical strength, comparable labeling. And there is one difference here, that it may differ in terms of the excipients. Because every manufacturer is making it differently, it may not have the same excipients. And one of the questions that a lady asked in the morning, that it had two different products, had a completely different dissolution profiles. In one case it was soluble, it dissolved fast. In the other case it was a different medium. That might be due to the excipients. Depending upon what excipients are used in that generic manufacturer compared to the innovative product, it may explain at times as to why there is a difference in the dissolution profiles. And therefore it may differ, again, different shapes, size, and all that, as long as the other things are maintained. Now, this was all with respect to the pharmaceutical equivalence. Now comes the bioequivalence, and that's the aspects we discussed in the morning, how a bioequivalence study should be carried out. But there are 4 distinct ways how a bioequivalence measurements can be done. The most common one is the first one, which is the in vivo measurement of the active moiety in the biological fluids. That is the pharmacokinetic measurements, which is true for most of the cases, and that's being followed. Now, in some of the cases, as you will see it, it is not possible to measure the drug concentration in the plasma, or the methodology is not existing, or the route of administration is such like topicals that you cannot measure the drug in the biological fluid. So what do? Then we follow what is known as the pharmacodynamic measurements if they have it. So you follow the pharmacodynamic measurements. If that's also not feasible, then you have the comparative clinical trials. And in some cases, even we use the comparative in vitro dissolution profile as a way of measuring the equivalency between the innovative product and the generic product. So these are the 4 different ways how we go about measuring the bioequivalence, how we go about measuring the equivalency, the methodology for those products. Now this is the same thing as I had in the previous slide, but because some of the products, like the topical products, you cannot measure the blood level concentrations, there was a change in the regulation in 2003, which said that for a drug that is not intended to be absorbed into the bloodstream, the secretary may assess the bioavailability by scientifically valid measurements to reflect the rate and extent to which the active ingredient or the therapeutic ingredient becomes available at the site of drug action. So that's again a principle that's being used in some of the topical drug administrations that can be easily followed. So it's a slight modification of what was done before. Now let's take a look into what are the locally acting drug products. The first one is the GI acting drug products, which are orally administered, but they are not absorbed, and they are meant for the local GI action. The second group of the products are those which are orally inhaled, and nasal drug products such as the metered-dose inhalers and dry powder inhalers. The third category is the topical dermatological drug products such as the semi-solids, which is creams, ointments, and gels for the local action. And some examples of the semi-solids are the glucocorticoids, antifungals, anti-acne, and these products. So these are the different products for which a different set of criteria is being utilized for the product to be determined to be a therapeutically equivalent or an bioequivalent product. Because remember, for a product to be therapeutically equivalent and interchangeable, it has to be pharmaceutically equivalent, which is very easy to do because you know the type of the dosage form, the strength, the formulation, the route of administration, everything is very easy, and you follow that. The most difficult aspect is how do we determine the bioequivalency of those products. So let's take a look into the locally acting GI products. Some of the examples are sucralfate. This is used for the treatment of the GI ulcers. And for these types of products, what is required is the comparative clinical trials. There is no substitute for that. And even though it has been approved several years ago, the only current requirement for the bioequivalency is the comparative clinical trials. The second set of products is the mebendazole. This is an anthelmintic agent which has the local action in the parasites. Again, for this, there is a need to do the comparative clinical trials. In addition to that, it also requires the pharmacokinetic studies because a small amount of the drug is still being absorbed, and especially with respect to the food, so you have to do the comparative PK studies with the food, and also you need to carry out the in vitro dissolution studies to make sure that the different polymorphic forms of the product are there, but they are all behaving in the same way. So these are again the requirements for the product like mebendazole. Also, there is a other group of products like cholestyramine, and that is a cholesterol lowering agent. And these products, you require what is known as the in vitro binding studies with the bile acids and the bile salts, and in vitro kinetic binding studies in the simulated intestinal fluid. SIF is simulated intestinal fluid in the presence of different concentrations of the bile acids and bile salts. So again, that's a requirement in terms of the cholestyramine, where no in vivo studies are needed, but these are the series of in vitro studies that needs to be carried out. Now we go towards still some more modifications of these, and these are the current thinkings right now, what the agency is thinking about it, like for the immediate release dosage forms, which are highly soluble drugs. For example, vancomycin. If that product contains qualitatively and quantitatively, Q1 and Q2 stands for qualitative and quantitative amount of the inactive ingredients. If they contain the same Q1 and Q2, then bioequivalency could be demonstrated by the in vitro dissolution test, which covers the physiological relevant pHs. In other words, in general, if you do the pH from 1.2 to 6.8, like 3 different pHs, then that should be okay. But if Q1 and Q2 are different, then in vitro and in vivo studies with respect to the pharmacokinetics and the pharmacodynamics, all the clinical studies needs to be demonstrated for these types of products, for vancomycin, for the bioequivalence determinations. Continuing on the immediate release dosage forms, the previous one, this is for the highly soluble drugs, and now this is for the poorly soluble drugs. That's where it is indicated that you can carry out if the Q1 and Q2 are the same, then you carry out the dissolution in a biorelevant media, plus there is a need for doing the in vivo studies, and this goes into the fact that what we were talking earlier in the morning during the discussion period, that the new products which are coming up are now more and more water insoluble or sparingly soluble or the poorly soluble drug products, and that needs some more additional requirements. And here the additional requirement is to do the dissolution in a biorelevant media, which may be containing some surfactants or some other materials. There are several publications with respect to what is a biorelevant media. We can discuss that later if needed, plus doing the in vivo studies. And if there is a modified release dosage form, then of course all modified release dosage forms do require in vivo and in vitro studies. So in summary, the locally acting drug products, GI products, in order to demonstrate the bioequivalency, in common the requirement is the clinical studies. Sometimes the PK studies are needed primarily to make sure about the safety aspects, and the alternate methods are there, such as for highly soluble drugs, the dissolution in the GI media, or poorly soluble drugs, the dissolution in the biorelevant media. Let's go to the second group of the products, which are again topically administered in a sense, for which these are the products for which there is no in vivo pharmacokinetic measurements feasible normally, and we do the studies. And again, there are two types of the products here. One is the bioequivalence of the nasal sprays. For these type of products, such as the solution nasal sprays, in vitro tests are enough for the bioequivalence determination. And if there are suspension nasal sprays, then you carry out the in vitro test to characterize the droplet size, aerodynamic particle size distribution, what are the different distribution size, and this can be measured using the instruments like cascade impactor or new generation impactors, measure the clinical equivalence and the pharmacokinetic studies to demonstrate the equivalency of the exposure. The direct measure of the particle size equivalence requires that if the drug particle size distribution of the test and the reference product can be demonstrated to be bioequivalent, which is not easy to do, then the nasal spray suspensions could be treated like the nasal spray solutions, and then there may not be need to do the in vivo studies. But that's not very easy to do in terms of demonstrating the equal particle size distribution because of the variations and the variabilities in terms of the measurements of the particle size. And for the bioequivalency of the inhalation products, then the oral inhalation or the MDI products, you require the bioequivalency through the in vitro testing for the device performance, because it is composed of a device and a material. So you need to perform the in vitro testing to make sure that the two devices are equivalent, carry out the pharmacodynamic studies of the lung function, of local delivery, and also the pharmacokinetic studies for the systemic exposure, because once it goes into the lungs, some part of it will be absorbed and it will be going into the bloodstream, and you do need to measure that. The same thing is true for the dry powder inhalations. The products, you need to identify the key formulation devices and the performance variables and do the bioequivalence studies for these products. So again, as you see it, it's not that easy to do the bioequivalency of the inhalation drug products, and in general then people end up in doing, unless it is a solution, a clinical studies. Moving on to the topical drug products. In the case of the topical drug products, the key step for the drug absorption is the release of the drug from the formulation, because it is only 0.5% or 0.05% of the active ingredient in a cream, ointment, or the gel. So it's just like a dissolution of the product. So first thing you need to do is what's to be determined is the drug release from the formulation, and then the drug penetration through the stratum corneum. Stratum corneum is the top layer of the skin, which is the dead layer of the skin, but the drug has to penetrate after it is released from the formulation into the stratum corneum, and then it is the permeability measurement, which is a rate limiting measurement, because if the drug is not able to penetrate through the stratum corneum, it will not have any activities. And then the thermodynamic activity is the driving force for the drug absorption, and the formulation can alter the barrier properties of the skin at times depending upon what type of formulation it is. Different types of the formulation, it may change how the stratum corneum is going to behave, and that's an important factor which needs to be determined. So these are the factors that we need to keep in mind when we are trying to measure the bioequivalency of the topical drug products. Again, just like the case with all the other types of the generic products you are looking at, one thing is certain, that product has been studied by the innovator and has demonstrated the clinical safety and the efficacy of the product. So that's an important factor. But then now, to define the bioequivalency as far as the topical drug products are concerned, is that it has to show that there is no significant difference in the rate and extent of drug absorption. That is the definition of bioequivalency from the FDA regulatory point of view. You have to show that there is no difference in the rate and extent of drug absorption. Even for the oral drug products, you measure the AUC and the Cmax, which is again the two important parameters, as you heard in the morning, for the determination of the rate, which is the Cmax, and the extent, which is the AUC. So again, those are the important factors which we have to keep in mind. The rate at which the drug can leave the formulation can be also an important factor to be determined, and the rate at which the drug can cross the stratum corneum, that is the permeability, is also an important factor. So these things are kept in mind when you are trying to determine the bioequivalency of these products. The topically applied semi-solid contains the corticosteroids. The steroids are the ones which have the vasoconstriction or the skin blanching activity, and that is the one which is used for the measurement of the bioequivalency of the products. We have guidelines on that, and I'll show you that in a minute. For the antifungal products, right now the requirement is comparative clinical trials, and the same thing is true for the anti-acne, that the comparative clinical trials. These are the requirements. For pharmacodynamics, the skin blanching is carried out, and this is the reference where you can get the FDA guidance on measuring the skin blanching. Now in skin blanching, as many of you may be knowing it, when you apply the corticosteroid, after some time it becomes pale. There is a vasoconstriction activity going on, the blood circulation gets reduced, and that's why the skin becomes pale. And this is more easily visible in the individuals who have a fair skin compared to the individuals who have the darker skins. And exactly how to carry out and how to do is identified in the guidance. And if anyone is interested, I will be happy to talk to you later on or during the discussions. Then the second aspect, again, for these topical drug products is the clinical studies, which is a fallback method for everything. When it fails, do the clinical trials. Well, because clinical trials are the gold standards. I think as Dr. Hinkal had a slide, or Dr. Ikar, I don't remember. I get mixed up between the two, just like everybody else gets mixed up. Who had the slide which said that this is the gold standard? Somebody used the word. You used it. I'm sorry, Dr. Rita. You used the gold standard. So just like it's a gold standard as far as the topical drug products are concerned, all the dermatologists feel that that is the gold standard and that's the way they would like to proceed further. But the problem is, its endpoints are highly variable. It is least accurate. Even the dermatologists will agree with that it is least accurate. It is least sensitive. It is least reproducible. It's an expensive, and it requires unnecessarily human testing. In spite of all this, it is still considered as a gold standard. But we as a scientist then have to start looking at some of the different alternative methods, how we could do that and how it could study. And therefore, that's what I mean by saying that there is a need for alternative methods to assure the bioequivalency and the drug product quality. So what are the alternative methods? One method which we had studied very extensively, and it is now coming back again, is the dermatopharmacokinetic method, where you measure the drug concentration in the skin, in the stratum corneum. You take the skin layers and try to measure how rapidly the drug is penetrating and how rapidly it is disappearing, just like measuring the plasma. It's a different way of doing it, and you can do that. And when this method was initially finalized and discussed, at the FDA, we always used to go and have a workshop where about 200, 300 people will participate, and then we will finalize the guidance or we'll develop the guidance. And again, that was identified in the workshop report which came out in 1998 about the DPK. It also identified the two other methods. One was the dermal microdialysis, which is being used now also. Again, these are all the trials where people have used it, and also the in vitro drug release from these topical preparations. So talking about the in vivo test, then the in vivo test quantified the effect of the formulation on the stratum corneum. First is the dermatopharmacokinetics. It measures the drug concentration in the stratum corneum. Second one is the microdialysis, which measures the concentration that have reached the dermis, because microdialysis is a technique where you insert a tube below the skin, and it is in the dermis area, and whatever the drug has gone from your stratum corneum inside below that, and that is in the dermis, and that's where the microdialysis technique can measure that. And it has been recently used for the evaluation of the bioequivalence of the products. And one limitation of that one is that technique, the microdialysis, is not appropriate for the drug which are targeted in the stratum corneum. For example, the antifungal agents, they are in the stratum corneum itself. The fungi resides in the stratum corneum, and therefore microdialysis is not appropriate. But then looking at the DPK or the dermatopharmacokinetic technique, that's the most suitable technique for the measurement of the antifungal activities, because the fungi are in the skin and you are measuring the drug the fungi are in the stratum corneum, and you are measuring the drug concentrations in the stratum corneum. You are measuring the drug at the target site itself. So again, that's an advantage of the DPK and disadvantage of the microdialysis. Talking about the in vitro release also has been studied extensively using the vertical diffusion cell system. It measures the diffusion through the formulation and the fraction of the free drug using the synthetic membrane. The reason why the synthetic membrane is used is it makes it easier, it's easily available, it can be easily reproduced. This is the same type of the cell systems that most of the manufacturing units, when they are developing the topical formulations, use it, but in those cases they use the skin, cadaver skin membrane. We have replaced the cadaver skin using a synthetic membrane here, but also it's important to remember that it is not predictive of the bioavailability. Though it is predictive, it's sensitive to the formulation differences. If there are two formulations which are different, or the same formulation has the different particle size, or it is manufactured differently, this system can easily detect that, but cannot be predictable right now for the bioavailability. Whenever you are looking at the topical formulations, you need to be aware of the rheology of the preparations, because sometimes it may have the same compositions, but the way it is manufactured could be different, and therefore the rheology of the product is very important, and it may be very useful in terms of determining the pharmaceutical equivalence of the two products. So taking a look into, again, how do we classify these topical drug products? The Q1 and Q2, as we saw in the other cases, Q1 stands for the qualitative amount of the inactive ingredients, and Q2 stands for the quantitative amount of the ingredients. But when these two ingredients are mixed in a different fashion, just like you get the different dissolution profiles depending upon how hard the tablets are pressed or what is happening, similarly it could be different here as to how the two cream preparations are prepared, and that could be easily checked out or seen from the Q3 measurements, which is the measurements of the drug release profile. So that's how the drug release profiles using the vertical diffusion cell systems, at least right now, comes out to be very useful and applicable. So then some of the questions which has come up in terms of evaluating these types of the dosage forms is the rheology the same between the two products? Is the adhesion to the skin is the same or different? Those are the things, again, it could be addressed through the in vitro drug release. Is the solubility of the drug in the formulation the same? Again, that needs to be addressed, and it could be identified using the in vitro release. Same thing if are the excipients released at the same rate, and is the particle size the same? Again, all this could be easily identified with the in vitro release. One important factor is there can be two formulations, because on the skin, if you see it, there are some hair follicles. So depending upon the size of the particles, one formulation may be more easily adaptable for the drug to go through the hair follicles and give you the different results compared to the ones which has a slightly larger particles. It may not be able to go through the skin and may give you a different result. So the follicular penetration, or the penetration to the hair follicles, is an important aspect that needs to be considered, especially when the product, when the active ingredient is in the form of a suspension. And that's where again the Q3, or the rheology of the product, or the in vitro release, comes and helps you to identify if the two formulations are the same or different. Now, it is this principle of the Q1, Q2 is being now utilized, or it's under a very higher level of consideration, I would say, because it is not yet finally approved, but these are the thought processes which are going on now at the agency, that if the product is an antifungal agent, which means it is targeted to the stratum corneum, then if Q1 and Q2 are the same, then you do the in vitro testing, and there is no need to do the in vivo testing, because of the in vitro, they are Q1 and Q2 the same. But if Q1 is equivalent Q and Q2 are different, the quantitatively, if it has slightly different amounts, then you carry out the in vitro testings. And in vivo test, if Q2 differences are potentially significant, again, you have to identify, knowing the ingredient, what is a potentially significant difference between the two. And if Q1 and Q2 are different, then you do the in vitro testing and also do the in vivo testing to demonstrate that there is no formulation effect on the drug absorption. Again, this is a thinking saying that, okay, there is no need to do the in vivo testing for the antifungal drug products if it meets the Q1 and Q2 criteria. And in most of the products in US, the generic products have the Q1 and Q2. The DPK, again, as I said, initially it was all developed, but then there were some issues, and now it is coming back again, and probably it will be coming back in a slightly easier format, easier manner than how it was done before. Before, we had about 8 samples taken, 4 samples to measure the drug uptake, and about 4 samples measuring the drug elimination, and now they feel that maybe just 1 uptake and 1 elimination will give us the more or less the same data, and that's what it is being used. But in most of the cases, again, we use the 90% confidence interval calculations for these purposes. Summarizing then, the locally acting drug products, methods for determining the bioequivalence. The bioequivalence studies with the clinical endpoints. It is recommended for locally acting drugs using the product-specific endpoints. But when we do that, these studies have dichotomous endpoints. Either it passes or it fails. And it requires the large number of subjects. The method, as we discussed earlier, is not very sensitive. It is inexpensive. And therefore, an alternative method has to be selected. And these are the other methods like the in vitro drug release aspects is used for the antifungal drug products. It is also used very extensively, at least in the United States, for the SUPAC-SS. SUPAC-SS stands for the scale-up and post-approval changes. After your product is approved, you make some more changes in your formulation, maybe with respect to some of the inactive ingredients, or changes in the manufacturing process, or changes in the manufacturing site. These are called the scale-up and post-approval changes, which in Europe it is referred to as the variations, and S stands for the semi-solids, which is the creams, ointments, and gels. So everyone now follows these guidelines, SUPAC SS for these kinds of changes. And again, it says that when Q1 and Q2 are the same, the only difference is in the manufacturing process, and that could be easily picked up by using the in vitro drug release. In conclusions, the determinations of bioequivalence for the locally acting drugs is a challenge. It's not as easy as the orally administered drug products where the drug is absorbed and it goes into the blood levels and you measure the pharmacokinetic aspects. Most of the products require the clinical studies. For the GI products, the in vitro binding studies for cholestyramine. For the other products, the dissolution plus the PK for the safety and the clinical studies. These are the, in brief, the requirements. For the nasal and the inhalation products, in vitro equivalence tests and in vivo PK/PD are the clinical studies. And for topicals, Q1 and Q2 are the same. Then the in vitro release itself, otherwise maybe DPK or the microdialysis or the clinicals. Right now it is the clinicals, but the DPK microdialysis is under evaluation process, so I cannot say that they are the finals, but looks like eventually, gradually, that might be the way we might be headed. And I would like to thank you